The plan now for the next several lectures, haven't actually counted, is to start looking at how neurons interact with information. There are lots of functions of neurons. They have metabolic functions, they have space-occupying functions, lots of things that they do. But the most important function in terms of their uniqueness is their capacity to process information. And so what we're going to do is start looking at how neurons process information within the cell of the neuron, and then talk about how neurons process information between different neurons and get progressively bigger and bigger until we finish up with looking at a system where the information is processed in steps through the whole system. Before we get into that, I want to spend today looking in a very pure physiological sense at what the functions of an integrating centre are, or what the features of an integrating centre are. An integrating centre can be something as small as, a tiny little nucleus that we can just about see, or as large as a cerebral cortex. It's just a centre where neurons are, where they process information. And all of those centres, however big and however small, have features of information processing. So every system that you will see has several regions that carry out different kinds of information processing. So that the sensory systems will pass their information on and on up a system, and the different areas or different integrating centres within that system will carry out specific processing on that information so that they work out what's going on in the world. And there are identifiable pathways that link the different components of the systems. So that as we get larger and larger, we can draw more and more complicated and improbable diagrams of different lines of axonal pathways or white matter tracts that connect different areas of grey matter to build this neurological system. And typically, neurological systems are diffuse throughout the central nervous system, by which I mean that we don't get one part of the brain that's all about motor function with lots of different nuclei for motor function and a completely different area that's all about sensory function. We tend to find that the pathways run through each other and around each other. That's handy in terms of function because it means that different levels of complexity of information processing and action tend to sit together. But it's complicated for us when we're looking at it because it means that when we're considering a particular system, there might be a tiny amount of grey matter down here that connects by a white matter tract to a tiny amount of grey matter right over here that's doing that processing. And so it's a difficult scale for us to interact with and work out what's going on. Nevertheless, the pathways, although they're complicated, they do project in an orderly manner. And we can see examples of the order of that projection as we look through the central nervous system. The last two things to say about the systems is that they are hierarchical. They take information, they do a bit of processing on it, and they pass that information up the pathway. And the other thing to say about the systems is that they decussate. Decussation is a big neurological word which means crossing the midline. Neurological systems tend to cross the midline. So when we look at the principles of organisation, we tend to see something that looks a bit like a computer. This is not a whole computer, this is a video card from a computer. And so, as a whole, this piece of computing plastic, or whatever it's made of, has a particular function to do. This particular piece of processing unit is made to generate a video image which comes out here and appears on your screen as something that you can interact with. In order to do that, it has an input It has a processing system and it has an output. And all neurological systems are the same. It has specific bits of plastic or chips or grey matter which do particular jobs which contribute towards that processing. And those particular bits of plastic are joined up by multiple pathways in a hopefully logical manner between them. And so we've got there a unit of processing that's made up of smaller units of processing. And I guess most of us know that right at the heart of all this is a series of more transistors that you can count, which are either on or off all the time. But when we look at the complex functions of a neurological system, we don't tend to address those in terms of the transistors. Okay? When you're surfing the internet or watching a video on YouTube or using, a word processor to write an assignment or something, how many of you are thinking about all those little transistors that are flicking on and off inside the computer? Anybody? We're interacting with that machine at a much higher level. And although what we see on the computer screen, we know is due to lots of transistors flipping their states way on down in the bowels somewhere, it's too much like hard work to think down through all those stages. Yeah? In just the same way, when we consider neurological systems, it's too much like hard work to think about all those excitatory postsynaptic potentials and inhibitory postsynaptic potentials as we go along. So as we consider bigger and bigger systems, we tend to think in more and more abstract ways about them. And so you'll see that as the systems get bigger, I'll be talking less and less about individual neurons and more about the kind of processing of information that goes on. But you need to try and remember that it all pins down to those individual synapses and changes in membrane potential that we talked about in the first few lectures. We just don't draw that line very often because it's a really complicated process to get from one end of it to the other end of it. In very abstract terms then, this is the basis for all of the processing that goes on in the nervous system. There is an integrating centre, a thinking centre, a processing unit, which generates a motor output at some point in some form. And that motor output does something. And then there is a sensory system which senses what's happening around, both in the environment and in terms of the output of the motor function, and it feeds that back into the integration processing unit so that the unit as a whole can integrate what's going on in the environment and the way the animal is interacting with the environment into future motor outputs. So, for example, if you're running for a bus and you perceive the back end of the bus getting smaller, your integrating centre will say, run faster, you fool, you're going to miss the bus. Okay? It's all about changing what you're doing to make your behaviour appropriate for the environment that you're in. And we call all of that feedback. We'll come back and talk about feedback in more detail in a moment. But for the moment, this interaction between the motor system and the sensory system that's at the basis of all our neurophysiology is called feedback. Okay, what then are the properties of these integrating centres? What properties do we see in these centres that are features of the way in which they function and which allow them to carry out their function? Well, these are some of the more important properties and some of the ones that I'm going to go through today. And you will see that some of them are inherent properties of all integrating centres and that some of them are properties which some integrating centres show. For example, delay is a function of all integrating centres. You can't process information without instituting a delay in the transmission of that information. But you can't be both convergent and divergent at the same time. So some integrating centres show convergence and some show divergence. a Golgi silver stain of a single lower motor neuron in the spinal cord. A dye is injected into the neuron, and all of these processes are all the dendritic processes of that one neuron. So this is its dendritic tree, the area from which it gathers information. The pathways that affect the function of this lower motor neuron attach to it via synapses on this dendritic field or on the actual body of the neuron itself. And for a single lower motor neuron, there can be somewhere between 50,000 and 200,000 synapses converging onto that neuron. All of those altering the final information output of this lower motor neuron that's innervating the skeletal muscles. So this is a convergent pathway. Many pathways are converging down to a single or to fewer pathways. And in general, convergence is a feature of the motor systems. The motor sides of our neurophysiology tend to be convergent. Information comes down from many places in the higher centres of the central nervous system and converges down on this lower motor neuron, which is integrating all that information to decide on a final output. The opposite of convergence is divergence. And divergence tends to be a feature of sensory systems. This is an illustration of divergence in the midbrain of the visual system. You can see that the optic nerve is coming down and through the optic chiasm into these brainstem centres, which you will have met, being the rostral colliculus and the lateral geniculate nucleus. And within those structures, the information from the optic nerve diverges and goes to lots of different places within the central nervous system. Some of the information goes up to the visual cortex and allows us to see consciously. But other pathways take the information down to the motor nuclei in the brainstem that control the external ocular muscles and make the eyes point in the right direction, and the parasympathetic nucleus in the brainstem that controls pupillary diameter and so lets the right amount of light into the eye, and the cerebellar cortex and the tectospinal tract, which adjust our posture and the muscle tone in our postural muscles so that we assume an appropriate posture in line with the horizon, and the ascending and descending reticular formations, which are concerned with arousal and which adjust our state of arousal. So you can see here that we've got a divergence of this sensory information going to various places in the central nervous system where different aspects of that information are useful. We see that with all of the sensory systems tend to be divergent. All neurological systems impose a delay. The delay is due to the time it takes for the action potential to propagate along the axon or the neurite, and the time it takes for the information to cross a synapse. And I think I said in the first two lectures that information travels in neurons at somewhere between about 0.5 meter a second and 120 meters a second, depending on the neuron or the axon, and that it takes about a millisecond for information to cross a synaptic junction. So there's delay. This is illustrated here in the EMG, the electromyogram, of a professor of surgery who allowed me to stick needles in his leg and then to tap his patellar ligament. You guys will all be familiar with the patellar reflex, where you tap the patellar ligament and the knee jerks up because quadriceps femoris contracts. If you measure the voltage generated by the muscles over time, starting at the time where you actually tap that tendon, you will find that the muscle is electrically silent for, in this case, 24 milliseconds after the tap. And that 24 milliseconds is the delay imposed by the information going up the sensory nerve to the spinal cord, across the synapse in the spinal cord and back down the motor nerve to the neuromuscular junction, across the neuromuscular junction to the muscle fibers, and also the time it takes for the electrical change in the muscle fibers to cause a mechanical change and a contraction in the muscles. Okay, we're getting electrical changes here at about 24 milliseconds. The mechanical changes, if I measured them, would come on a little bit later. So that delay is inherent in all processing systems. Fatigue, or adaptation, is also a feature of not all but most neurological systems. This is a graph of action potentials, or rather it's a graph of the membrane potential with action potentials superimposed of an axon in the optic nerve. And it's showing the way the rate of action potential generation changes when we shine light on the retina in the area that this axon is relaying information from, in the sensory field of this axon. You can see that initially when you shine the light on, the rate of action potential generation increases. So we get a faster generation of action potentials from the basal rate. But as time goes by, we get fatigue in that pathway, or accommodation, and the rate of generation of action potentials tends to change back to its baseline rate. And then when we turn the light off this area of retina, we get a reduction in the rate of action potentials, and then that fatigues back to its baseline rate. So most sensory systems have a baseline rate of action potential generation which changes when we alter the sensory information to that system. There are 3 different kinds of feedback in our motor systems. The first one is ballistic control feedback. This is one of the British archers at the Olympic Games, and her cunning plan is to shoot this arrow into the middle of a target some 90 metres away. And generally speaking, archers at the Olympic Games are very good at that. Once she's loosed the string and the arrow's gone, she's got no way of controlling where the arrow goes until the next time she shoots an arrow. Yeah? Nothing that she does after the arrow's gone is going to affect that arrow's flight. So what she's doing is getting the desired result, arrow goes into the middle of the target, and looking up stored programs in the motor system that the controller thinks will deliver that result. Generating a motor output, and then the motor output, together with any noise in the system, like the way the wind's blowing that day, or if there's any humidity in the air that slows the arrow down, giving us an actual result. The actual result feeds back into the stored programs so that next time she shoots an arrow, the controller looks up in the adjusted programs and gives her something which is more likely to generate the actual result. So learning takes place over a series of iterated attempts to do what the archer's trying to do. It's called ballistic because in the same way as throwing a ball, once it's gone, you've got no control over it till the next time. The other main kind of motor feedback is continuous feedback. And this time, the actual result is continuously monitored by a sensory system or comparator that feeds information back to the controller so that the controller can adjust the motor output on an ongoing basis to alter the motor output as it happens. So in this example here, this gymnast is trying to perform a very flash maneuver on the rings called a crucifix. And the tension in the arm muscles and the sense of balance and posture generally are feeding back to the control system throughout the maneuver. so that the gymnast can adjust the motor output as the manoeuvre's going on to improve the result. So it's like a continuous feedback control system. The third kind of feedback is a bit of a cheat because it's not feedback at all, it's feed forward. And this is the situation where the controller uses its experience of the situation to decide what's going to happen in the future to plan an appropriate motor output. So that, for example, if somebody throws you a ball, you look at the flight path of the ball and you adjust yourself and the position of your hands so that they're in the right place when the ball comes to you. You're looking at your sensory information and planning what's going to happen in the future to line your motor activity up with that. When we talk about facilitation, it's worth mentioning Sir Charles Sherrington, who won a Nobel Prize in 1932 for his work on facilitation, and so became known to be the grandfather of modern neurophysiology in doing that. And it's worth talking about why his work was so special. At the time Sherrington was working, there were a lot of people who were looking at the nervous system to see what it looked like. People like Golgi, whose stain I showed you a few slides ago, who were saying, these are what the neurons look like in the central nervous system, this is how they arrange themselves. And doing some really beautiful and detailed studies, and for the first time showing where neurons went to and how they got there and what the structure was like. But if you ask people like Golgi what those neurons did, they wouldn't have had any tools to be able to give you the answer. And at the same time, there were people like Pavlov, who we'll meet in a few minutes, working, who were interested in the functions of the central nervous system and interested in the ways in which the central nervous system learned and adapted its activity to the situation that it found itself in. But if you ask Pavlov which neurons were doing the learning that he was talking about, he wouldn't have had the tools to be able to tell you. So the anatomists like Golgi had no way of telling you what the neurons they were looking at were doing, and the functional people like Pavlov had no way of telling you which neurons were doing the functions that they were beginning to identify. Sherrington came along, and for the first time he said, here's a change in function in neurons, and it's those neurons that are doing this change in function. So for the first time, he was able to marry together structure and function in the intimate way that we understand it to be so important for neurophysiology at the moment. What he did was, he looked at a group of neurons which had a pathway coming out of them and pathways going into them. Convergent neurons, as it happens in the spinal cord. And he stimulated input A and got an output A. And then he stimulated input B and got an output B. And then he stimulated, excuse me, A and B together, AB, which gave him an output AB. And what he found was that the output AB was greater than the sum of the output of A and the output of B. What he hypothesized from this was that these are not just two different neurological pathways, each going out about their own business and not interfering with the other one at all. What's happening is stimulation of output A, of input A, is providing an output, but it's also facilitating, and that's his word, facilitating other neurons, which means that they don't necessarily contribute to the output, but they are brought closer to their threshold, and with another stimulus, they are more likely to undergo an output. So that when you stimulate A, It may be that you stimulate these 3 neurons here and facilitate neurons in a curve around it. And when you stimulate B, you stimulate these 3 neurons and facilitate neurons in a band around it. So this neuron here is only responding when A and B stimulate at the same time, because it's facilitated, though not completely stimulated, by both. And in this model here, output A is 3 neurons, and output B is 3 neurons, but output AB is 7 neurons, because it's the 3 neurons each, plus the one that's facilitated. And so he demonstrated, again for the first time, this interaction between different groups of neurons that he called facilitation. But just as importantly for modern neuroscience, he was able to say, these are the neurons in the spinal cord that are providing that function, and led other workers who followed him to start looking more and more closely at the interactions between structure and function. Summation is another feature of neural centers, and summation is a wider term that includes facilitation. Facilitation is a form of spatial summation, in the sense that these neurons are close to each other, they're related spatially, and we get summation when we stimulate more than one pathway in that area. Temporal summation is the same kind of thing, but it's a change over time. If you get an excitatory postsynaptic potential on a neuron that changes the membrane potential towards threshold but doesn't take it above threshold, so doesn't generate an action potential, that postsynaptic potential hangs around for a period of time. And typically those postsynaptic potentials hang around for around about 30 to 50 milliseconds or so, for a considerable amount of time in a neurological sense. And during that period of time, those neurons are facilitated and open to being stimulated by other pathways. And so if 2 signals arrive in a similar time frame to each other, such that one facilitates and one arriving later causes stimulation, that is temporal summation. The different signals summate over time. Inhibition is being shown more and more to be probably the most important function in the cognitive centers, in the higher centers of the nervous system. And if you think about it, that makes sense. If all of your pathways were excitatory, and you had a large network of mutually excitatory pathways, and you gave it a stimulus, then that stimulus would just excitate more and more pathways and get bigger and bigger like an avalanche, until by the time you got the whole thing locked into this excitatory sort of, I don't know, catatonic state, you'd have no idea what the original stimulus was. Yeah, so a stimulus to your left toe would induce a general state of excitation, and a stimulus to your right toe would do the same thing. And you wouldn't, in the higher centers, have a chance of being able to distinguish between the two. So inhibition tends to keep a lid on this excitation and keep pathways shut down and limited to the size of response that is appropriate. It limits their responses in terms of space over the central nervous system. It limits responses in terms of time after the stimulus. And it's a necessary and important part of all of the neurological networks that we see. And of course it's generated through not excitatory postsynaptic potentials, but through inhibitory postsynaptic potentials. I want to look now at lateral inhibition, which is a special case of inhibition which is prevalent through the sensory systems and which is extremely important in understanding the ways in which our sensory systems convey information. I've drawn here a diagram of an area of skin with a number of Pacinian corpuscles in it. And I've simplified their sensory fields as hexagonal areas of skin, just because hexagons tessellate and they join up with each other and it makes life easier. So here we have the sensory fields of different sensory Pacinian corpuscles over an area of skin. And what I want to do now is to look at the output in terms of rate of action potential generation in those different sensory areas. And you can see from this tasteful pink that I've coloured it in that the rate of action potential generation is at a standard sort of baseline level throughout the area, where this sort of red colour is an increased rate of action potential generation and the white colour is a decreased rate. So all of these areas are generating action potentials all the time at a standard rate. So what happens now if we take a feather or some other similar device and we stimulate just one of those centres in the middle by giving it a tickle? What's going to happen to the rate of action potential generation in these sensory fields? Well, the answer is, and I've drawn the diagram a little bit larger here, the rate of action potential generation in the sensory field that's stimulated increases, and the rate of action potential generation in all the sensory fields around it is inhibited, it decreases. So we get this lateral inhibition or lateral decrease in output from areas adjacent to areas that are stimulated. Might seem a bit odd to have 7 sensory fields changing their output just because one of them was stimulated. But if you look at what happens when we stimulate a larger area, so we tickle over the whole of this area, you'll begin to see why that's useful. If I draw the same diagram of increased and decreased rate of action potential generation for this area, it looks like this. And if I take the T's away to make it clearer, you can begin to see what's happening, I hope. In areas well away from the area that's stimulated, there's no change in action potential generation because these areas are neither stimulated by stimulation or inhibited by stimulation in any of the areas adjacent to them. In the middle of the area, there is also no change in the rate of action potential generation. But this time it's because these areas are both stimulated by direct stimulation and inhibited by stimulation of all the areas around them. When we go out to the margins, we have areas just within the area of stimulation, but on the edge, which are stimulated directly, but not completely inhibited because some of the areas around them are outside the area of stimulation. So they increase their rate of action potential generation. And if we look at areas which are adjacent to but just outside the area of stimulation, we have reductions in action potential generation because this is not stimulated directly, but it is inhibited by stimulation in these areas around it. So can you see that we're building up a picture which looks at the edges of the area of stimulation, that demarcates the borders of stimulation? And this is useful for 2 reasons. One, it gives us a better spatial resolution of the area of stimulation because we get this contrast between increased rate of action potential generation and decreased rate of action potential generation. And 2, as the area gets larger and larger, the number of areas that are altering their rate of action potential generation and so effectively sending information to the central nervous system increases linearly with the increase in diameter of this area of stimulation, whereas if the whole of the receptive field that was stimulated responded, the increase in information would be the square of the size of the field. because it's related to its area and not its circumference. So what we're doing is we're getting better spatial resolution of a stimulus and transmitting less information to the central nervous system. So we need fewer neurons to process that information, and that's less overhead in terms of metabolic activity to keep those neurons alive. So it's a better system and it's a more efficient system. Reverberation is a feature of some neural networks, usually the neural networks associated with nociception or pain perception. What tends to happen is the information, as it goes up the excitatory pathway, branches off and goes back down the pathway, and so you get a self-excitatory pathway. A very small stimulus here can cause a very prolonged and dramatic response to that stimulus at this point. And that response will carry on either until these neurons fatigue and stop firing, or until an inhibitory pathway turns them off. And as I said, we see that with pathways where, even though the stimulus might be small, it's a very important stimulus and the response needs to be large. So that, for example, if you touch a hot surface and it feels painful, you completely pull your hand right out the way, even though it's only a little bit of pain on the tip of your finger. You don't kind of go, ooh, that's painful, I wonder what's going on there. Oh, it's burning, I better move my finger, there we go. It's a much more pronounced response to a small stimulus, and it's generated by reverberation in those sensory pathways. Talked about Pavlov, who won his Nobel Prize in 1904 for, as I'm sure you're aware, linking novel stimuli with reflex responses to produce new reflex pathways. Yeah, Pavlov's salivating dogs. Everybody familiar with those? The point of this is that this is one of the first demonstrations of learning and memory within a neurological system. And learning and memory are features of neurological systems that we need to bear in mind. All neurological systems have memory and can learn, and they do it in particular ways that we'll address later on in the course. Somatotopic localization or somatotopic mapping. This is the concept that the localization of the tissues of the body from which sensory information comes or to which motor information goes is reflected in the physical localization of the nervous tissue that deals with those areas of the body. So, this is an area of cerebral cortex, and drawn above it are the areas of the body which relate to this cerebral cortex. And this little man figure here, which is called a homunculus, Latin for little man, is drawn in proportion to the area or number of neurons that process information relating to that part of the body. So that you can see this homunculus has a very large hand, because lots of information processing is going on there around the hand, and a relatively small armpit because we don't really pay a lot of attention to the information that goes to or comes out of our armpit. That somatotopic mapping goes on at all levels throughout the central nervous system and the spinal canal. Next time when we get together, we'll talk about the ways in which neurons communicate within themselves.